Second speaker is one of the rising stars of biomedical science at the McGill International TB Centre. Yves Dube is an early career researcher who won an award in biomedical research at the TB research event hosted by the McGill International TB Centre. And he's working on the basic science of innate immune response to Mycobacterium tuberculosis, and he's going to be talking today about pattern recognition receptors, immunity and susceptibility to Mycobacterium tuberculosis in mice. And I'd asked Yves if he could keep his talk to about 20 minutes, so we have got some time for questions at the end. Thanks very much, Yves.
Thanks for the introduction, Greg. It's an honor to present at your symposium. I haven't presented internationally, been invited to present internationally before, so this is very nice for me. Thank you. So yes, as Greg said, I'm a PhD student, PhD candidate in Marcel Baer's lab in Montreal, and I'm working on basic science focusing on mice and the genetics of these mice and how this relates to susceptibility to TB. So I'll get started here. First, I want to introduce you to a reagent that's used a lot in medical research. This is called complete Freund's adjuvant. It's basically mineral oil plus heat-killed Mycobacterium tuberculosis. And the mineral oil by itself, which is incomplete Freund's adjuvant, or IFA, is thought just to contribute to creating a depot effect for a slow release of adjuvant. Whereas for the complete Freund's adjuvant, with that contains mycobacteria, it's known to elicit cell-mediated immunity, which is the same type of immune response that you are probably more familiar with in a tuberculin skin test than someone who's been previously exposed to Mycobacterium tuberculosis. So when Jules Freund was developing this adjuvant back in the 1950s, he remarked in a review that he thought it was unlikely that the same factors in Mycobacterium tuberculosis that influenced the immune response in this adjuvant would have no role during the infection that they cause. So we thought about this and thought that maybe we can approach this and question and with modern technology, and we formed this hypothesis around this, that mycobacterial factors that alter immunity do play synergistic roles during tuberculosis. Two of these factors are the cell wall peptidoglycan of mycobacteria and fragments thereof. So on the right here, this is the muramyl dipeptide fragment peptidoglycan, which is recognized by the host pattern recognition receptor NOD2 located inside host phagocytes. And historically, the cell wall is thought to be the key ingredient in complete Freund's adjuvant for its ability to elicit cell-mediated immunity. Another of the factors that contribute to immune response to mycobacteria is the cord factor, which is in the mycomembrane, so the outer membrane of the mycobacteria. And this molecule is recognized, particularly, this is the minimal motif that is recognized by the Mincle receptor located on the surface of host phagocytes. And what we know about cord factor is that it's sufficient to generate granulomas, at least in mouse models, which is a characteristic pathology of mycobacterial infection. So one of the models that we use to address this Freund's adjuvant-driven immunity is this mouse model where we immunize mice against either complete Freund's adjuvant, which has the mycobacteria, or incomplete Freund's adjuvant, which is just the mineral oil. And in the presence of ovalbumin, just as an example antigen that we can then re-stimulate T cells with later to measure the immune response. And we used various mice. So we had our regular C57BL6 wild type mice. We had also obtained Mincle knockout mice, NOD2 knockout mice, and Mincle and NOD2 double knockout mice, which we obtained by crossing the Mincle and NOD2 knockouts. So in this way, we're addressing the factors of mycobacteria and their importance by having mice with and without the receptors for these factors. So these mice were immunized against ovalbumin with our adjuvants. We waited 7 days and then harvested draining lymph nodes and re-stimulated these cells with ovalbumin ex vivo to measure the antigen-specific immune response. And this is a remark about the origin of these mice. We obtained the Mincle mice generously provided by Christine Wells in Melbourne, which we would have been trying to obtain these mice for about 18 months prior to that, derived from sperm. But luckily she had started a colony of her own, and we were able to ship live mice from Australia to Canada. Without this contribution to study, these studies could not have proceeded. So just a thank you to Australia. Okay, so one of the results we had was that we saw that NOD2 and Mincle do contribute to cell-mediated immunity from Mycobacteria and CFA. And so what I'm showing you here is the frequency of interferon gamma producing CD4 positive T cells in the draining lymph nodes after we had immunized mice. First, you'll notice that in any one of these graphs, you can see that unstimulated cells have a very low background of interferon gamma production because they don't have the cognate antigen ovalbumin to which the mice were immunized. Next, you'll notice that the CFA immunized animals have the highest antigen-specific response compared to the higher antigen-specific response compared to the IFA immunized animals. This is the mycobacterial contribution to cell-mediated immunity that we're interested in. Next, if we look at mice who are lacking either NOD2, Mincle, or both NOD2 and Mincle, we see an approximately 30 to 50% decrease in the immune response compared to their wild-type controls, indicating that these pattern recognition receptors contribute fractionally to the immune response to mycobacteria. So this is with front adjuvant is with dead mycobacteria. So we're also interested, of course, in how the immune response behaves when the mycobacteria are alive, because of course we know TB has different factor proteins in it that allow it to evade or alter control the immune response of the host to it. So this is just a simple macrophage infection in vitro. So these were bone marrow derived macrophages from the mice of these various genotypes. On the left panel, this is heat-killed Mycobacterium tuberculosis, pipetted onto these macrophages and then we looked at the immune response in terms of TNF alpha produced in the supernatant after an overnight incubation. And what you can see quite clearly, there's a genotype-dependent immune response to these mycobacteria, similar to what we see in the adjuvant model. And then if we look at live Mycobacterium tuberculosis, we quite clearly see that this is also true during an encounter with live Mycobacterium tuberculosis, in which case, compared to wild-type mice, a certain amount of the immune response is lost in Mincle knockout mice, and then a greater amount is lost in NOD2 knockout mice. But interestingly, that NOD2 Mincle, NOD2 double knockout mice seem to phenocopy the NOD2 single knockout mice, suggesting that in the absence of NOD2, there isn't much of an importance of Mincle to the remaining immune response. So to take this a little bit further, instead of just macrophages, you want to look at these, the role of these pattern recognition receptors in live mice. So we have in our containment level 3 facility here at the Research Institute of McGill University Health Center, we have an aerosolization device. So we are able to infect mice through an aerosol procedure. So just to go through it briefly, we secure mice in a plastic conical cylinder with an opening for their nose to stick out, and their nose is exposed to an aerosol stream for 15 minutes, which consistently gives us approximately 30 colony forming units or CFU of mycobacteria in the lungs as measured 24 hours post aerosolization. And so then after this, depending on the experiment, we either monitor the mice for weeks or months until they reach an endpoint, which is either a time point or a point for euthanasia. And then we harvest different organs to measure different things. So first we wanted to look at the ability for bacterial control in these lungs, in these mice, in the lung, spleen, and liver as sort of a simple way of assessing how important are these pattern recognition receptors for the control of a mycobacterial infection. So we looked at approximately 3 and 6 weeks post-infection first, and what you'll notice is that though these graphs are on log scale, but there doesn't seem to be a large difference between the different genotypes in these organs when we're looking at these are medians. So in the lung specifically, we see about approximately 2 to 2.4-fold increase in the burden of bacterial burden compared to the wild type control, although this is not statistically significant, especially because of the multiple comparisons. In the spleen, we see what looks like maybe be a larger difference at 3 weeks. There really doesn't seem to be a difference at 6 weeks, but this is also not significant, and similarly with the liver as well. So there may be a small role for these pattern recognition receptors to play in bacterial control, but it's certainly not striking like you would see in an interferon gamma or TNF alpha knockout mouse. We had also looked at lymphocyte numbers and as well other cells in the parenchyma of the lungs at 3 and 6 weeks at the same time as we looked at CFUs. And what we see is there seems to be a trend to reduce T and B cell numbers in the lungs at 3 weeks, but this also completely disappears by 6 weeks. So it's possible that there is a slight delay in the mounting of the adaptive immune response in these mice, which would be compatible with the adjuvant data that we have before, where we see that cell-mediated immune response in terms of T cell responses are really dependent, at least partly, on these receptors. So there might be in live infection a delay, but by 6 weeks, the mice look quite normal in terms of lymphocyte numbers. So we also had done a survival experiment as well, because we want to know when these immunological and bacteriologic parameters don't show us something, we want to understand whether these pattern recognition receptors are important for survival during TB infection. So we had started this experiment in the beginning of 2020. So it took a little longer than we expected. It took 1 year to complete, but it was just not very abnormal for the TB mouse model. You typically would get survival times, depending on your experiment, between 3 months, 6 months, or 12 months. So this suggests that these knockouts are also leave the mice relatively immunocompetent. They're not really sick like an interferon gamma or TNF alpha knockout. So what you'll notice is that, so the median survival times of the Mincle, of NOD2 knockout and Mincle NOD2 double knockout mice were very similar, around 290 days post-infection. And then the Mincle single knockout mice was a little bit after that, around 310 days post-infection, if I remember correctly. And the experiment had ended at 363 days before the wild type had reached a median survival. And looking at the statistics of this, we see that the NOD2 and double knockout mice seem to be statistically significant. Mincle knockout mice were not significant, correcting for multiple comparisons against the wild type. So the last piece of data that we had taken from these mice was H&E and ZN stained lung sections upon euthanasia. So these sections were taken from the survival experiment when mice were euthanized, so not at a specific time point, but that this is the pathology associated with death from tuberculosis in these mice. So this is a wild type mouse. You can see that in mouse TB, we don't have clear granulomatous structures. We get a sort of a diffuse pathology and consolidation of the lung that spreads throughout the lung until there is very little airspace left for the mice to breathe. Usually the mice are euthanized before that point. So you can see in wild type mice, there are some areas of lymphocytes collecting. There are some areas that are left relatively healthy that still have airspace. What we had noticed in mice that were lacking the NOD2 receptor was that where we had seen a lot of consolidation of the lung tissue, we noticed these necrotic lesions, necrotic foci rather, that would appear in a few but not all of the mice. So just to take a look a little bit more closely, I'm going to zoom in on this part of the lung. And what we see is on the top of the screen, there are necrotic cell debris. And then around this is surrounded by a cuff of foamy macrophages, and it's adjacent to the consolidated tissue around it. And this is also associated with a lot of acid-fast bacilli. So in these necrotic lesions, this is a place where Mycobacterium tuberculosis seems to be quite happy and growing unabated. So in summary, 3 of the 12 NOD2 knockout mice had these large necrotic foci, and 2 of 14 double knockout mice had these large necrotic foci. So a minority of the mice, but we had only observed them in these. So then we didn't observe them, it could have been that mice were euthanized a little earlier based on their body score, and maybe we hadn't let them get sick enough yet. Maybe this might be a very phenotype that's very close to the dying of tuberculosis. So to conclude about what I've shown you, we know now that NOD2 and Mincle contribute to cell-mediated immunity to Mycobacterium tuberculosis, whether this is in the form of live infection or dead bacteria in an adjuvant. And that double knockouts seem to phenocopy one or both of the single knockouts. So this to me suggests a sort of a synergistic response from these pattern recognition receptors, and that in the absence of one receptor, it looks lacking. Also, the other receptor makes no difference, because without one, the signaling of the other just becomes unimportant. And also so the absence of these receptors seems to result in accelerated mortality, but it's at a late time point. So there is a role for these receptors to play in tuberculosis, but it occurs very late. So the phenotype seems to be very rather mild compared to other knockouts like interferon gamma or what have you. And the NOD2 knockout seems to resemble a double knockout, whereas the Mincle is intermediate. So this also this could be that NOD2, you are more likely to die of that the NOD2 phenotype occurs faster, or again, that this could be maybe a synergistic phenomenon. In the published work, we showed that these pattern recognition receptors and their ligands can be used as, their ligands rather can be used as adjuvants, and that they work synergistically together in cell systems and in adjuvant systems. So if you combine Mincle and NOD2 agonists, this actually makes an adjuvant that works quite similarly to the whole Mycobacteria in CFA. So these two molecules explain approximately 50% of the adjuvant activity of Mycobacteria. And more philosophically, that sensors like these seem to be dispensable for control of the bacteria, which is very different from interferon gamma or TNF alpha, which are effectors. So the way we're seeing is that we have a lot of pattern recognition receptors, and in the absence of one or even two, maybe even 3 or 4, the host still has a lot of sensor molecules to detect the presence of Mycobacterium tuberculosis, and that the difference in the host's outcome with lacking 1 or 2 or a few of these receptors will probably manifest as something more subtle and maybe immunological in nature. So this is, we think there's a redundancy in pattern recognition receptors, and that if we're looking at the role of genetic control in human populations with variations in these pattern recognition receptors, we should probably be looking at something subtle like an endophenotype, and not just whether someone becomes has active tuberculosis or doesn't have active tuberculosis or converts their TST. Might be some that we have to look at something a little bit more subtle. So I'd like to thank everyone for your attention, for inviting me to this symposium. It's very exciting for me. And of course, I'd like to thank my supervisor, Marcel Baer, for providing me with this project over the last few years. It's been very interesting, and I'm looking forward to publishing the last pieces of work on this. And as well as I'd like to thank the people in my lab that have made this possible, because a lot of these hours in the containment level 3 facility, and as well as work upstairs, it takes a lot of hands and a lot of help. So they're invaluable to me as well, current and past members. I'd also like to thank our collaborators and the technological platforms at our institute, as well as our funding agencies here in Canada. And thank you again for your attention.
Thank you very much, Jean-Yves, and congratulations on a very impressive body of work. We have some time for questions, so we'll pass the microphone around. And as I said before, if you could introduce yourselves and where you're from, that would be very helpful. Thank you.
Oh, hi, Jean-Yves. This is Jamie Trickus from the University of Sydney. Thank you very much. I just have a question about the fact that you saw this increased mortality at this extended time point. Do you think that that would also relate to greater bacterial loads at those longer time points? Is that something you're looking at, or is it other things in terms of inflammation, even though the bacteria are controlled?
So we did, I mean, it's hard to plan an experiment to take a 9-month time point with mice, because like you don't know how many mice you'll have alive. But what we did do was, when we euthanized the mice, we had taken the lungs for histopathology, but we have livers and spleens that I had looked at, enumerated the CFU, the bacterial burden in these organs. So this is it's not a specific time point, but it was the bacterial load in the non-lung organs associated with death from TB. And what we see is, if we plot this on a time axis and do a linear regression, we can see that at least in the double knockout compared to wild type, there does seem to be a slightly increased bacterial burden not in the liver rather, but in the spleen. So there does seem to be a slight loss of control of bacterial loads. It's within 1 log, I think. So it's mild, and we don't know if that's really the reason the mice are dying. It seems that the lung pathology is more important. But yes, it is possible that there's a slight loss of bacterial control.
We have a question from online from Catherine Aronica. The NOD2 knockout mice appear to have many cholesterol clefts in the lung. Do these mice have a metabolic phenotype as well?
Yeah, we had talked to a histopathologist about that, because we didn't know what those things were, and he told us they were cholesterol. But I haven't quantified it yet, but we do see that also in the wild type mice. It would be a matter of, these slides we've gotten relatively recently, so we haven't finished analyzing them. But yeah, the cholesterol is present in wild types as well. But we haven't quantified the difference yet. There might be a quantitative difference, but it's not obvious.
Okay, thank you very much. So again, could everyone join me in thanking Jean-Yves for his presentation? Thank you.